Hello everyone, I am delighted to present our recent research paper published in the iMeta, titled Maternal Gut Microbiota-Derived Daidzein Prevents Osteoporosis in Female Offspring Following Prenatal Prednisone Exposure. The DOHD theory highlights the enduring impact of early life adverse exposures on offspring's long-term health and disease susceptibility. Osteoporosis is characterized by high incidence rates and severe clinical consequences. However, its pathogenesis remains incompletely elucidated. Emerging evidence reveals osteoporosis's developmental origins during intrauterine programming. Prednisone, as the primary oral glucocorticoid for managing maternal conditions during pregnancy, manifests a well-documented therapeutic paradox. While providing clinical benefits, prenatal prednisone exposure demonstrates significant developmental toxicity. PPT/PPE induces long bone dysplasia, reduced PBM, and increased osteoporosis susceptibility in female offspring. Maternal gut microbiota metabolite DAI mediates PPE-induced osteoporosis susceptibility in female adult offspring. DAI mediates the inhibition of osteoblast differentiation and increased susceptibility to osteoporosis in PPE offspring via KAT6A-mediated epigenetic regulation of HOX12 expression. DAI emerges as a target for the early prevention and treatment of PPE-induced multi-organ functional changes and related disease susceptibility. The retrospective case control clinical study demonstrated that PPT induces fetal femoral dysplasia in female fetuses and alters maternal gut microbiota composition in the human cohort. Using a PPE rat model that mimics clinical prednisone dosing regimens, we demonstrated that PPE induces maternal gut microbiota dysbiosis and causes long bone dysplasia in female fetuses. Analysis of femoral bone mass in female offspring rats at postnatal weeks 12 and 28 revealed that PPE resulted in reduced PBM. Maternal gut microbiota transplantation experiments demonstrated that gut microbiota dysbiosis induced by PPE mediates the reduction in PBM and impaired osteogenic differentiation in female offspring. Maternal DAI supplementation during pregnancy significantly reversed PPE-induced reductions in PBM and impaired osteogenic differentiation in female offspring. Transcriptomic sequencing of fetal bone tissue combined with postnatal local HOX12 overexpression experiments demonstrated that reduced HOX12 expression mediates PPE-induced PBM suppression in female offspring, establishing HOX12 as a key downstream effector of PPE-induced osteogenic dysfunction. Animal studies demonstrated that DAI reversed PPE-induced downregulation of HOX12 expression. In vitro experiments further revealed that HOX12 mediates the DAI-enhanced osteogenic differentiation of BMSCs derived from PPE-exposed female offspring. PPE-induced persistent reduction of H3K9AC levels at the HOX12 promoter region. In vitro studies further revealed that DAI upregulates HOX12 promoter H3K9AC levels and enhances its transcriptional activity through KAT6A-dependent mechanisms. Molecular docking and in vitro experiments demonstrated that DAI promotes KAT6A expression through binding, thereby increasing H3K9AC levels at the HOX12 promoter and enhancing its transcriptional activity. DAI enhances KAT6A expression via binding to thereby increasing H3K9AC levels at the HOX12 promoter and upregulating its transcriptional activity. PPT induces maternal gut microbiota dysbiosis and suppresses fetal bone development in female offspring. Animal experiments further demonstrated that PPE causes long bone dysplasia, impaired osteogenic function, reduced PBM, and heightened OP susceptibility in female offspring. Mechanistically, maternal gut microbiota-derived metabolite DAI mediates PPE-induced long bone dysplasia and PBM reduction in female offspring by KAT6A-dependent epigenetic programming of HOX12 expression during prenatal and postnatal osteogenesis. Maternal DAI supplementation during pregnancy reversed PPE-induced osteogenic function, low PBM, OP susceptibility, and developmental impairments in multiple organs, liver, hippocampus, adrenal glands, and ovaries. DAI as a novel therapeutic target with translational potential for preventing fetal origin OP. Furthermore, maternal supplementation of microbiota metabolites emerges as an effective preventive strategy against glucocorticoid-induced fetal developmental programming disorders.